Welcome back, everyone. So the next session is on open science, and I think Anke, in her opening remarks this morning, framed this session beautifully when she said that as soon as you embraced open science, people who work in your field are no longer necessarily competitors, but they can be collaborators. And I think it's a nice summary of this session. So this doesn't necessarily crystallize rapidly, and forming these communities, it can happen slowly. So the question is, first, how to catalyze this formation of building these communities, and this is what Albert will be telling us first. And second, what are mechanisms to bring these communities together on specific topics, specific projects that are obstacles in the field, and bringing this community of people together on specific challenges. And this is what will be the object for the second talk. But so first, so the first speaker for this session is Dr. Albert Antolin, who is a group leader at IDIBELL ICO, in Barcelona. So he's an organic chemist by training. And then he was for 2 years in silico drug design scientist in the pharma industry. After which he moved back to academia at the IRC in the ICR, in the UK, at Columbia University, and then he returned to Spain to start his own lab. His own lab that is interested in developing open science, chemical biology resources, harnessing the effect of drugs at a systems level in precision medicine, and discovering new cancer drugs leveraging big data and AI. So since 2024, and I think that's what he's going to tell us about today, he chairs MAINFRAME, which is an international open science network of machine learning scientists and computational chemists dedicated to advancing pre-competitive drug discovery research and promoting open source AI method development. So please join me in welcoming Albert. Yeah. How are you?
Cheers. So is this? Hello. So thank you very much for this kind introduction, and to Conscience for inviting me. And yeah, I hope you'll join us in this adventure that is MAINFRAME. So to explain why we've started MAINFRAME, I thought I'd go back a little bit. And one kind of example that it's a bit, kind of influenced MAINFRAME is the Human Genome Project. So as you all know, there was a competition between a company of Craig Venter and a public consortium, and the public consortium kind of won the race. And I think this has been good for everyone, including industry, to have this pre-competitive available of the reference human genome. And another thing that's kind of interesting, if you think about, and Al already mentioned a little bit of this, but 25 years after the Human Genome Project, we still don't know the function of more than 30% of the human proteome. And we continue to study the same group of privileged proteins largely. And I think the SGC has been great over and over, demonstrating that having chemical probes available to study understudied proteins really boosts and increases our understanding of these proteins and opens the door to new drug discovery on those proteins. So what do I refer to when I talk about chemical probes? So I thought it would be nice to make sure we are all in the same page. What we talk about when we talk about chemical probes is not just inhibitors, but these compounds need to be really specific and have other properties to make sure that when we use them to study the biological function of a target, we are really measuring this target, right? And this brings me a bit to this kind of initiative called Target 2035 that was also started by the SGC that aims to bring all the community together to try to discover a small molecule chemical probe for every human protein by 2035. Okay, so how many probes or chemical probes do we have? So for this, I'd like to bring your attention to this resource that I have the privilege to contribute to, the Chemical Probes Portal, which is really a huge initiative, and it's a curated database and web resource of chemical probes that I would encourage everybody to use, particularly when you want to find a more selective inhibitor for your protein of interest. So again, you can query the website, you can find, as I said, the more selective inhibitor for your protein, and this is all human curated. So you have reviewers that have assessed those compounds, and importantly, will point you to the strengths and limitations of these compounds, right? So you can use them with knowledge of what their limitations might be. Sorry. But this also enables us in this kind of curated space to know for how many proteins do we have chemical probes for. And the reality is that despite a lot of effort of these human curators and reviewers, and as you see, it's increased a lot over the recent years, so this is a huge effort, but we have chemical probes for around 570 human targets. So it's not a lot. So are we missing those probes? No? So another thing I was involved with that it was really nice initiative was try to measure, to identify probes using data science. So what we did back when I was in London, and of course London, a lot of rain. So we had this vision in the Department of Data Science that we had to bring a lot of different data together to make decisions about target prioritization in drug discovery, but also we were able to do interesting analysis like the one I'm mentioning. So we brought together genomics data, pharmacological data, perturbational data, chemical probe data, expression, et cetera, in a huge database called CANSAR. It's a relational database, and we've published about this before. So the nice thing is that we can now use this relational database to make other analysis and interesting connections between this data. So what I did as a postdoc back in 2018 was to use this data to objectively assess chemical probes at scale. So we use CANSAR, and then I developed 6 chemical probe scores that measure some of the key properties of these chemical probes, like, of course, potency, but also selectivity, cell potency, SAR, inactive analogs, and paints. And we packaged all of these in a hopefully user-friendly website we call ProbeMiner. So again, you can search this by target, and you are prompted with the more potent, selective, etc., compounds available for your target of interest. So the cool thing about having all these huge databases is, of course, we can now analyze things at the proteome or human proteome scale. So for example, another nice thing of CANSAR is, of course, we have AlphaFold inside, and we had developed a machine learning method to predict drugability based on pockets. So we can now assess with AlphaFold for all the proteins in the human proteome, how many of them we predict that are amenable for small molecule modulators, that they have pockets that are likely to be druggable. So I don't know if anybody wants to guess, but in our hands it's 42%. So 42% of the human proteome, so around 8,500 proteins, are potentially ligandable. And this is kind of a very conservative measure, right? Because now we have PROTACs and other forms of modulation, so it's likely much higher, because we don't need canonical binding pockets for some of these new modalities. But again, I don't know if anybody wants to continue guessing how much of this we have identified ligands for. But this starts dropping quite rapidly, because if we look at Campbell, how many human proteins have a known ligand below 10,000 nanomolar of affinity, it's about 16%. So again, all of this is kind of opportunity, targets with potentially druggable sites that have no known ligand whatsoever. And if you go a bit further and look at how many of those liganded targets have a known small molecule drug, well, it's very, very low. It's interesting to check and compare with the number of human targets that have probes, because it's also around 3%, but the target spaces don't completely overlap. This is quite interesting, and it's because for some targets, we have approved drugs that are not selective, if you think on multi-kinase inhibitors or GPCR drugs that are very unselective. And however, of course, for some targets that, for example, fail in clinical trials, we have selective inhibitors that are not probes. But anyways, so the idea of this plot is to show that chemical data exists for a very low number of human proteins. For the majority of them, we don't have any small molecule inhibitor known. We went even a bit further, and we, to make it a bit provocative, we tried to assess over time how we've been doing over the last years. So what you see here in green is different Campbell releases over the years, and how many human proteins have are ligandable, no? Have a known small molecule binder below 10,000 micromolar, 10,000 nanomolar, sorry. And as you can see, we keep increasing, which is nice, right? But we keep increasing very slowly. And in blue, you have the chemical probes that we identify in ProbeMiner that have what we call minimum quality. So again, we keep growing even more slowly on the probes. But if we think about target 2035, and in 10 years having a small molecule chemical probe for every human protein, we are not doing this fast enough, right? It would take us even 100 more years to get there. So I think the key thing, and I'm getting to the aim of creating MAINFRAME, is current technology, no? And clearly, current technology, what we are doing now, it's not enough. So I can pass through this very quickly because we already heard about cash challenges, but particularly from the first challenge that Matthieu explained, which was the one where data was very scarce. Current methods are not doing very well, right? So very few hits were identified, and their potency is very, very low. So I think it's very clear that we need to do better. So again, and a lot of experts from the SGC and industry got together and started to think how we could improve current methods and what were the bottlenecks. And they identified 3 main pillars, no, that would facilitate to improve these methods. The first one is data. So as we've heard, we don't have data for a lot of proteins, and the data we have is normally fragmented, like very biased, etc. So could we generate a lot of data to train better machine learning models? That's kind of the first pillar. The second one, and we've heard a lot about this as well, is challenges, or these kind of open benchmarks that enable us to not only to compare and find who wins a challenge, but to objectively compare algorithms under the same conditions and find out which methods work better so we can improve upon them. And finally, and we've heard a bit about this as well, community. We really need an engaged community that participates in these challenges, that uses this data, and that has this will to improve the models. So let's touch on this very quickly. So the SGC and a lot of pharma partners are starting a huge data generation initiative to screen over 1,000 human proteins with billion-sized libraries and put all the data in the public domain. So this is a very, very exciting initiative. And what I most like about this is not only that we are generating data orders of magnitude bigger than what we currently have, and that the data will be, organized full matrices of data, so things that we don't currently have, but also that this data is being generated with the idea of helping improve machine learning methods. No? So it's not the primary objective, it's not finding hits, but it's improving current algorithms. So this is a bit the roadmap, and there's a paper coming up very soon explaining this. But basically, purified proteins would be screened with 2 technologies that I'm going to explain in a second. And then we would run computational challenges on those proteins. There would be prospective validation, very much like cache challenges. And then all the data would make it to the public domain. And again, just to point you that there will be options as well for scientists to submit their proteins of interest to be screened if they can purify them. So there will be two main technologies, affinity selection mass spectroscopy, which is a mass spectroscopy-based technology to test binding of your compounds. These are relatively or slightly smaller screening libraries of 400,000 compounds, more or less. And then there's DNA-encoded chemical libraries, which are small molecules encoded with DNA tags. And the amazing thing about these types of libraries is that they are billion-size compounds. So we are talking about screening a lot of compounds against our targets of interest. And all these data will be deposited in AirCheck, and you have a talk on AirCheck tomorrow, so I'm not saying more about this, and Ben it's going to talk about this tomorrow. So, data. We are going to have data, as I said, orders of magnitude bigger than what we have now. The second idea is challenges. So as you know, and you've already heard this morning, we are talking about a diversity of challenge organizers and trying to help come together to run a diversity of challenges with this data. So what's coming initially will be hit finding challenges, because that's one of the primary objectives we have, so finding hits for proteins that are understudied and for which we don't have any information. But of course, we are going to generate a lot of other data, so there will be challenges as well on pose prediction, on congeneric series affinity prediction, and again, and this is coming through to MAINFRAME. So the idea again of this is to talk a lot to the community. So we are very open to have feedback and to organize challenges on your ideas. So for example, I was in Barcelona last week as well, and we were discussing with all INQUIS and other scientists that maybe selectivity was a very key thing that we should be running challenges on, right? So maybe we need to have several targets of the same family and running these DNA-encoded chemical libraries and trying to organize challenges to predict selective hits or non-selective hits. So again, this is very open for everybody to participate, and I hope you share our excitement of being able to run our challenges on this data. So the first challenge is coming up. Again, Gustavo already mentioned, but here you have the QR code. So I hope you join us in this first challenge. I think it's an incredible opportunity to join early with this. There's not a lot of DEL or DNA-encoded chemical libraries, machine learning methods out there in the public domain. So I think it's a very cool opportunity to join early in this adventure with us. So the idea of this challenge is that we will ask participants to train their models on DNA-encoded chemical library screening data that we have. The target will be known, so you could also apply structural data, structural methods, but the idea is that you also train on the machine learning. And the nice thing, or the challenge of this challenge, is that there will be another library of ASMS screening, which is a different chemical space from the DEL library. So, and what we are asking is that you build machine learning methods that are able to predict hits from the ASMS library, which is a bit challenging, but can be done. Here you have UMaps that we created of the different libraries. So in green, you have the ASMS chemical space, and in blue and orange, two different DEL libraries. So as you can see, there's some overlap in the chemical space, but it's not complete overlap, which make it quite challenging of these machine learning methods to predict across chemical spaces. So yeah, that's it from this challenge. I hope you join us. And there's a 5,000 cash prize if you need a bit more additional incentive. Oops. No, this stopped working again. Sorry. Now, finally. Okay, so the last one is community, and that's MAINFRAME, so what I've been talking about. So again, we will be data at a scale we didn't have before. So our desire is to maximize the use of this data. And learning a bit from Kaj and AlphaFold, we've heard about these examples before, but I think it's really key that we build a community around this data, build a community around these challenges in order to maximally use it, no? And again, one of the motivations we are trying to encourage is having open models out there that everybody can use, no? So the idea is try to encourage that, and I think as the human genome, the advantages are huge of having these open models available to everyone. So and then it doesn't prevent companies from then doing drug discovery. But it would be really helpful if you think also in neglected diseases, so diseases that don't have such commercial interest. So we started MAINFRAME in December, so not so long ago. And this is growing quite rapidly, I would say. So this was the map we had about a month ago, and this is today. So we are over 130 scientists from 35 countries. The idea is a network of machine learning and computational chemistry scientists to participate in the challenges. So we are trying to encourage participation on this network by giving some benefits that we think are interesting. So some of the challenges, what we are thinking now is that the first time we do a different type of challenge with a different type of data, it will only be open to MAINFRAME members, like to act as a bit of a beta testers of new challenges. As you saw with CASH, for example, there's a lot of effort to do not only retrospective validation, but also prospective validation. And this would be only free of charge if you are a member of MAINFRAME and your algorithm performs among the top. So I think this is huge benefit, no? Because sometimes I run a small lab and it's difficult to pay for this prospective validation. So I think this is a huge opportunity to validate our methods in real world applications. And then, as I said, so the idea of this network of people is that we influence as well data generation. So we'll have regular meetings as well, and we'll have the opportunity of designing challenges if we have new ideas, or influencing, for example, which chemical space should the libraries cover, and if we should increase, for example, the size of the libraries, etc. And then we will run regular meetings as well, and we will help winning labs to come to these meetings. And what we are asking labs that are members of MAINFRAME to do is to open source their models, because if we don't open the models, it's much more difficult to learn and which methods are working better. So this is an open source network. And we ask, of course, members to participate in the benchmarking challenges and come as much as they can to the meetings we'll organize to discuss the results of these challenges. This is the map, as you saw, and I think we are also trying to make an effort to make this global, a global network, and we have some already fantastic members, part of MAINFRAME. So Karina, you've heard already about her very good results in CASH competitions. So, for example, Marvelous Ajala, who's running a network of machine learning in Africa, for example, and we hope we will be able to grow there as well. Or Jose Medina Franco, who has been leading chemoinformatic networks in Latin America, for example. Or Huabin, who is our MAINFRAME coordinator and will help grow us, our network as well in Asia. And of course, there are many challenges that remain. We are a very young network, so we still need to grow in many countries where we are not present. We don't have a huge number of members from industry, so you're very welcome, and I hope we can grow there. And again, we've heard talks about this before, so I think it's very interesting that we start exploring ways to engage the community better, no? And we are, as I said, very new, but we will be setting up communication channels, etc. And we plan to do as well networking and training, etc., to try to bring the community together. And again, as I said, we are very open to new ideas in this space. And yeah, so I think the meetings to analyze the challenge results and discuss among ourselves will be a big part of this network, and we hope to start organizing them after the first challenge finishes. And that's it. So I hope I have convinced you that chemical probes are really key to help us understand the function of proteins that we don't know and kickstart new drug discovery projects. That data, challenges, and creating a community are the key ingredients to boost this research into new chemical probes. And I hope you'll join us. Let me finish by saying this Nature editorial, no? A couple of years ago, so for chemists, the AI revolution has yet to happen. And there was a letter opposing this saying the AI revolution for chemistry is not that far away. So I'm completely convinced of this. And I think if we if we put out there the data and the challenges, we can accelerate this massively. So just to finish, thanking my lab, funding, Conscience, and of course the SGC collaborators, and you for listening. Thank you.
And once you have these communities, I guess the next step is to invite them to offer them, if you will, some platform where they can actually work together, where they work and synergize and actually solve real world problems. And I think it could be the focus of the next talk, which is also super exciting, where we'll hear about Open Molecular Science Software Foundation, which is a platform where people can congregate to work on force fields, on free energy calculations, on protein folding, on ADME talks. So great diversity of real-world issues, problems, barriers. And to talk about this, so we are lucky to have Karmen Čondić-Jurkić, who is the co-founder and executive director of the Open Molecular Software Foundation. So she holds a PhD in computational chemistry, and her research was focused on studying protein function and dynamics using multi-scale modeling techniques. So during her postdocs, she developed passion for open science and research infrastructures. And she's particularly interested in building new resources and making research infrastructure more robust, sustainable, and accessible, and facilitating large-scale collaboration projects. Karmen?
Let's see if this is working. It is. Okay. So it's very nice to be here. Again, I would also like to thank the organizers for inviting me. Like, this whole event is kind of my jam and what I spent last, too many years, I guess, working in, probably about 7 years. But I guess my interest in open science kind of goes back in, I think, my first postdoc, which was in 2014, which seems so long ago. I'm revealing my age now, so maybe I better start talking about why am I here today. And the title of my talk is, Can we build roller coasters? Now, you must be wondering what the roller coasters have to do with, molecular modeling and open drug discovery. But at the time when I had to submit the title of my talk, I was reading one book very, very often. And I'm sorry, the title of that book, The Pigeon Will Ride the Roller Coaster. And that is a favorite book of my 4-year-old. And it's a very fun book, and I highly recommend it. And in there, in the book, the pigeon learns about roller coasters and obviously gets very excited, wants to ride on one, and dreams about riding a roller coaster and thinks about how it's going to be this amazing, thrilling, exhilarating journey. And he finally gets to the place, sits in the car, and then what happens to be that the ride is just a little train that did pat, pat, patty, pat, pat. And as I was reading this book every day, I was thinking I relate to this. A lot of time when I was doing research and I wanted to do something, and I would find a tool, and I thought, yes, this is going to solve all my problems. Only to 2 weeks later, I only partially got it to work. It's not doing exactly what I thought it was going to do, and I thought I was going to jump on a roller coaster, but it was just a train that did pat, pat, patty, pat, pat. And this is how, I guess, I got interested in how can we build better research infrastructure. I thought we're there a lot of very smart people, doing research. And then if they all have to spend 2 weeks just to make the basic tools work, to solve actually another problem, that's probably not the best use of their time. And I thought, okay, we must, be able to do something about it. And I was also still very young and also probably a little bit naive, and I didn't know what the wise pigeon knew, is that building and riding will not be easy. It is not, right? And so even though sometimes we are set to build a roller coaster, all we end up is a train that does pat, pat, putty, pat, pat. And maybe that sometimes, you know, because our dreams were too big, or maybe sometimes we hit some constraints that, you know, we weren't aware of before, right? And, you know, and I think this is also a good metaphor maybe for AI hype, right? Where people say, oh, this AI tool will solve all of the problems and you won't have to do anything else anymore, no more experiments. And, you know, that's the hype, I guess. And then the dream is that maybe the model is also sometimes just the train that does putty, pat, pat. It's also maybe a good metaphor for expectations, right? Like, let's say some people are adrenaline junkies, and even the craziest roller coaster will just feel to them as a train that does pat, pat, putty, pat, pat. But for some people, you know, maybe they're scared, and maybe even the, you know, pat, pat, putty, pat, pat train will be exhilarating journey. And sometimes, you know, we also have to like realize when it's appropriate to build a roller coaster and when, you know, also pat-pat trains are enough or good enough, right? And so in trying to, I guess, solve this problem of like how can we build better infrastructure? Or if we decide to build roller coaster, how can we do it, you know, jointly? How do we do it together? And so one of the primary, I guess, aspects of OMSF's mission, and so I would just like to say that OMSF is a 501c3 not-for-profit organization based in the States, but we are fully remote and globally distributed. I don't have a map, maybe I'll bring it up next time. But one of the primary aspects of like what we do every day is develop software and build new technology. But we are not developing like cutting edge things, you know, in some ways. We are kind of doing things that have been proven before, but we are trying to do them better now. For example, development of force fields, right? Because often tools that are developed in academic labs, once, you know, if you work in industry, you don't have the luxury of trying to get things to work in 2 weeks. You have to, you try 1 time, maybe 2 times. If it doesn't work, you don't use it, right? So it has to be, you know, things have to be robust, well documented, and, you know, they just have to work. And then another thing that that is important to us is access. So everything that we build is released under permissive open licenses. So it's easy to reuse or, you know, to build upon whatever we have released. And that includes both software and data sets we produce in the process. So if you want to use any of our infrastructure, any of our code or data, you are free to do so. And you are free to do, you know, to get really creative in how you want to use these things. And finally, we also want, not finally yet, but we also want to facilitate collaboration, right? Like if you want to have multiple institutions working together on joint projects, you have to facilitate it somehow because, you know, there are legal agreements involved, money changes hands, all of this like has to happen somehow. And obviously you have to coordinate with people to kind of come, you know, sit together at the meetings, decide what needs to happen, figure out what these joint interests are, and, you know, and also how to get it done. And finally, that's when we realize you kind of need an institutional home that can support such activities. Like you need a place where you can put money in, you need someone to sign the agreement, you need someone to be legally responsible for certain things. And it turns out that universities are not always the best places for these things because, you know, they have a fundamentally different mission, which is, you know, to educate new generations and support researchers who, you know, work there as PIs and, you know, other things, you know, but not necessarily support open tools for specific research domains. And I think that's fair. And finally, we want to be sustainable. And, you know, sustainability obviously means a lot of different things to a lot of different people. But, you know, I think I came to realize that it's usually money, whatever people tells you, you can buy. It can be volunteers, it can be community, but the most reliable way of actually sustain something is you actually pay for someone's time to work on a specific thing. Everything else is not very reliable. So the way how we raise our funds is through industry consortia. So we have industry participating and contributing through membership fees. So they decide which project they want to support and then how much they actually want to give. We are applying for grants. We are usually trying to partner with academics, so we reduce the burden of grant management, which is pretty heavy when it comes to federal funding. We are also experimenting with providing services. So for example, if you want to extend our any specific, I guess, tool or code base that's being developed at OMSF, maybe you want to customize it. talk to us and we can help you because we do have expert developers in-house. And we can also receive donations, but I think this is probably the least utilized revenue stream that we have at the moment. So I think that's barely worth mentioning for now when it comes to how we sustain ourselves. And then I would also like to go back to what Anke said in her introduction earlier today, is like how open makes some things easier and how competitors become collaborators. But it's also, there is a practical aspect to it. So when we sign up agreements with our industry partners, they all sign the same agreements, which says everything that's being produced will be released under permissive open licenses. And that saves actually a lot of time in legal negotiations. And anyone who has worked with lawyers know how expensive they are. So we are just saving money and time by, you know, treating everyone the same and saying this is how everything is working, and, you know, you can decide whether you're in or out, but there is no special treatment. Everyone gets the same, like, participates under the same conditions. And I think that's really important because it evens the playing field. Like, it just makes everything easier. It also makes it easier for us when we communicate to universities, right? Because they also have to, like, green light their researchers if they want to collaborate with us. And so overall, I think it solves some other IP issues, so we don't necessarily need to require copyright to be translated to us as long as it's released under permissive license. And I think that kind of does help us to bridge this gap between industry and academia and, you know, facilitate some of the collaboration. So it's not always, like, it's easy to say we really want to collaborate, but then if you really want to, there are like a gazillion of like problems to solve in you know, the real world, from like, which nobody wants to do, from legal to accounting and other things, right? And that can actually, you know, bring collaboration to halt, right? If those things don't work out as well. And so I'll just quickly go through how these OMSF projects work, especially with the industry consortia. So usually we have academics involved in some capacity, and it really it depends from project to project what's the level of involvement. But the model that we are kind of trying to adopt is that we see that how we approach it is that academics are kind of developing, like, you know, cutting edge tools and what we, you know, we refer to as prototyping, right? And so to go back to the roller coaster metaphor, like, it's okay if you're an academic and you develop a patty train. It's okay if things are not working. It's okay if, you know, your code is not the most beautiful looking, as long as it does what it's supposed to do scientifically. So you have proven that, you know, whatever algorithm or method you have done works. But then, you know, we have our core team of developers who are building up, like, you know, the core package of, like, the core functionality, and they also are kind of overseen by the industry advisory board, who kind of provide the input. What are the key features that they want to see developed? What's the most important thing that, you know, should be considered and placed on the roadmap? And they also participate in, like, there are monthly meetings, they provide feedback, they participate in the benchmarking studies, which are again released under permissive licenses. And the team, and, you know, and this is actually really the key, having a team of, like, dedicated people whose, you know, whole job is, you know, to do this work really further facilitates collaboration. Because if you have to, like, assign certain task or responsibility to volunteers that kind of come and go, that just becomes an intractable problem over time. So this also provides a certain level of stability. And so I'll quickly go through the projects that are currently housed at OMSF. So it all started with Open Force Field. Open Force Field was founded back in 2018. And just again to let you know, like, so this is what everyone wanted, right? And yet it took 18 months of legal negotiations with all the industry partners, like, who was setting up the consortium, right? And so after Open Force Field was formed, and, you know, the first results, you know, were promising, and everyone was kind of also excited about this new collaborative model, then Open Free Energy came about, and that was kind of the moment where we started to think, okay, maybe we can spin up a new entity that can actually support these projects. And then just a few months later, it's actually just as, so OMSF was founded in 2021, And then in late 2021, I think first version of AlphaFold was released, and then we got in touch with OpenFold people, and they were like, we want to actually set up an industry consortium because we are really worried about this model that's being developed by, you know, a big company. We don't know what it means for, you know, for the users, what kind of terms and conditions will come with using AlphaFold. And that was obviously much before AlphaFold actually released their model, like the first model under as, I guess, open source licenses. And there is this concern, right, like that people will be locked out or locked in, right, into something and maybe pay some exorbitant fees. And, you know, again, it's about access. So they set up a consortium around OpenFold, and they're about to release, I guess, OpenFold 3. Hopefully soon, but they are it's hard, right? Like it's a hard problem to solve, and they keep like stumbling into challenges, and I can maybe talk about it later. And then we have WESPA. So Open Forest Field, Open Free Energy, and WESPA are all like physics-based modeling. WESPA is about developing enhanced sampling methods. And our latest addition is Open AdMet, which is an ARPA-H-funded project in collaboration with UCSF and Octane Bio, where ARPA-H is funding what everyone is talking about, a generation of new data sets aimed at machine learning. And OMSF assists with building data pipelines, machine learning, and we will obviously also, not obviously, but we plan to launch a series of blind challenges, and hopefully in collaboration with Polaris and anyone else, I guess, who wants to join. But this is like, again, another like more AI directed project. And finally, we are also providing home to some community building projects, such as Open Rosetta. Open Rosetta is a branch of the Rosetta Commons for their educational activities, networking, and also a place for them to think about what comes next, right? Like as the Rosetta, because the new technology that comes out of the Rosetta Commons community now is not necessarily directly related to their, you know, legacy Rosetta package. So it's all also, again, more machine and AI oriented. And so they're also thinking about what it means for them as a community, and, you know, what kind of skills they want to invest in the new generations moving forward, et cetera, et cetera. We also have BPDMC, which is a shortcut for Boston Protein Design and Modeling Club, which is just a monthly meeting in Boston for people over pizza and beer to discuss the latest development in protein design and modeling. It's very cool. So if you're in Boston, you know, you should check out if there is an event going there. Alchemistry is another event about free energy, mostly still based on physics-based modeling, free energy calculations. And the virtual Winter School of Computational Chemistry is also the latest edition. It's a virtual event for people to get training in computational chemistry. It's as the name says, it's completely virtual, so people can join from any place in the world. It's free for participants and provides resources and like records the lectures, and anyone who participates can access it at any point in time. So it's a great, really, community resource. And there are multiple ways also for you to get involved. The hardest probably thing it would be to start a new project or consortium, or you can bring your own if it's aligned with our mission. You could also join an existing consortium, which if you are a company, you should consider it. That means that you could become a member and influence, you know, the roadmaps of these projects, and also get to collaborate with other people, other researchers from other companies, which I think they all enjoy. You can apply for grants for us. If you think that what we're doing aligns with what you want to do, you can reach out to us and we can see if we can also support your proposal. You can also just go to all of the GitHub repositories, contribute code, data, or compute, which is always, you know, we are falling short always on compute. And or you can join our benchmarking efforts or challenges that there'll be more of them coming up soon. Finally, if you want more lightweight engagement, you can come to one of our events. The next event is in Boston on May 9th. So if you're, again, in the neighborhood, come and join us. I think there are still a few tickets left, and the event is free, so you can just join and hang out with us. You can donate or sponsor, but let me know if that's what you want to do. I mean, we will be happy to take your donations, but the expectations are low, I think, at the moment. Or you can subscribe to our newsletter and follow us on our social media. That said, I guess whatever you decide to do, I think it's fair to remind you that building or riding a roller coaster probably won't be easy. Because what it takes to, let's say, build an open roller coaster. This is what people, when we write grant proposals, or when we have ideas about what we want to do, we say we want community-driven research platforms. It sounds great. Who would say no to it? But it's really hard to do. Like you have to coordinate among like really lots of people, and sometimes they don't respond to your messages. And I was just thinking of Dylan and want to say, hey, Dylan, thank you for organizing. And I was also one of the unresponsive people in this context. But what our community manager and I came to refer to it, you have to yell into the void quite a lot in hoping that someone will hear. And so you can, coordinate, start working out certain things, and, it takes a lot of time. That also sometimes, makes it harder to, to allocate your resources efficiently. Or sometimes you just like suffer because you are like remote and everyone is in a different time zone, and so like everything is kind of sometimes moving slower than it would if it was just you and 2 other people in the same room. And oftentimes when a lot of people are involved, you lose some clarity about what is, that you should be doing, what is the, the highest priority. It's like constant fight against entropy. It's nobody's fault. It's just not easy, right? And then people say, we also want modular interoperable tools. Sounds great. Nobody wants to say no to it. But it's also really hard. Like the more flexible your tools you want to make, like the more edge cases you have to take into account. Like the more, whenever you try to make a more flexible solution, you take on more complexity that you have to deal with, right? Like the easiest thing is to say yes or no, but when you start like yes if this or yes, like it's just like starting to branch out and grow into like a lot harder dependency or complexity diagrams. And then there's also a maintenance burden. Again, if you want to pick one specialized tool that only does one thing, that's also easier to do than if you are trying to make it like make an interface with other tools or other outputs, and so it's hard. And then it's also obviously responsibility and credit. We want these projects to be governed by the community, but sometimes it means like really asking a good amount of their time to volunteer for us, and it is a big ask. Sometimes it's almost like taking on another job. For our board members, they're also taking legal liability on, so there are not like little things that we sometimes ask of people when we ask them to, be a part of the community. And finally, open access to both code and data. Again, sounds wonderful. That's what I thought when I was like 10 years ago. We should all do it all the time. The problem is like when you share things, and we could all have an S3 bucket on AWS somewhere and drop our data and make it publicly available, but it's not useful. In order to, make your data useful once you put it in the public domain, you have to annotate it, you have to curate it, you have to let people know what's inside, you have to let people know where to find it. Like, there are a lot of like generalist repositories nowadays, and they, fulfill a role, but oftentimes you can't find anything there unless you know what you're looking for. And like a lot of research is just like not really knowing what you're looking for. Like it's like looking around it and hopefully that you'll somehow circulate around the thing that you are hoping to find. So it's again one of the things that's easier said than done, and to say we should all make things open, but it's really hard. And like working in open and trying to even document and share what you have done, it's like additional overhead on. So not just that you have to do what you are supposed to do, you have to document it, you have to put it online, you have to really make it accessible to others and understandable, right? And as you can see, this image, I think it has a clear AI signature. I did prompt it, I think this was Gemini, and I asked it, can you make some roller coaster protein image? I don't remember exactly what the prompt was, but this is what it came up with. I also asked ChatGPT to help me with my slides. I told it, this is my title. These are like the bullet points that I would like to talk about. And And I also asked it, can you make an image of yourself as well? And it came up with this cute, helpful, I don't know, telesupport icon. And what was really nice from the response I got from ChatGPT said, that's a super fun and intriguing title. I love the metaphor of roller coasters. It suggests excitement, complexity, and the collective effort to build something bigger than ourselves. And I was like, yeah, I subscribe to this. And I was thinking, and it's so nice that it says I came up with, an intriguing and super fun title. I was like, oh, that's really nice. And, at the same time, I find myself often when I'm prompting, I sometimes put please because that's how I would ask like a colleague. And I type a lot like in Slack because we are fully remote. And, I know how my colleagues look like, but it's what I guess what Derek said in the morning, sometimes they really feel like you are talking to a person or, so I think we have to like pay a lot of attention, right? About what, so it feels like they are smarter or they understand. So I think it's really important to keep in mind what these things are, because they do hack us at some psychological level. But what was amazing, I guess, that based on my 3 bullet points and a title, it came up with this set of slides. Unfortunately, when I download the image, it just cropped it. I couldn't download the whole thing. I don't know why. But it came up with, pretty good story. I could present this easily. I think it would be fine. And I guess it's my own fault that I chose to go down and, give myself more work to do. And, it has some really interesting things, but it also makes some really interesting spelling mistakes, like transparent tools, the whole future with building, seamless integration, and collaborative lobs. I'm still to figure out what that means. There's also an interesting and alternative spelling of tools. I'm again not sure how to pronounce it, but overall I think it gets the right ideas, right? And the thing that kind of got my attention the most was this slide, the AI coaster is coming, right? And where we are warned about black one box, data blasts, and proprietary pitholes. And I agree with all of the things. Except I disagree that AI coaster it's not coming. It is here, right? Like it's not just about what AI will do. Like can we build another AlphaFold 3 or an AI tool that predicts ligand poses or ADMET or something. The fact that these LLMs are here, I think, will profoundly change how we do research. Like you can I don't know, put a data set into LLM and ask it, hey, can you slice it and dice it this way? Can you maybe plot this for me? Like, you don't have to do these things yourself in the same way, you had to until very recently. This thing that you could just prompt things into images, I think it's only a few weeks old. So we just did not have this until very recently, right? And it makes mistakes, but at the same time, it's amazing what it can do, right? Like, it's pretty amazing. And now it also does coding. And again, it makes mistakes. But what does it mean for us, like for people who are in software development? People are already losing jobs there. I think we're safe still because everything we do is a niche, right? Like, as again, referring back to Derek's talk, like where it makes mistakes are probably like all the edge cases. And this is where science is, right? Like it's a lot of edge cases. But even in science, there'll be like some roads well traveled, right, where we can, get good outputs and reliable ones. And so what it means for us, what does it mean open source, like if you can generate code on demand? Like, what does it mean for open source? Because open source was also like, fulfilling a role of being something that's, you can access for free. Maybe you pay in time because it will take more time to set something up, but it was there, and now maybe you can get your custom solution, provided that you have access to an LLM. But maybe it will mean accelerated discovery, smarter simulation and predictions, and again, potential for democratization, but also exclusion, right? what is open molecular software in the future, right? If you can code anything you want, and now I'm not saying again that the current tools can do any of these things, but if we kind of extrapolate, and even if they can do like only, do coding well at 50 or 60%, there will still be like boilerplate cases where they will be very useful, and and maybe they can at least, they can also just, improve productivity. We just have to work out in the same way for AI for research, when these models are working well and when they are not, right? But what it means then to be like open, like if molecular modeling software is now heading like towards AI direction, and this is the trend that we see. So everyone needs more data, everyone needs more compute. Is that something that will be accessible to everyone? If we think about then open source as not just like a license that you attach to your code, but like more as a framework, like access to resources, and for some time that was access to software. And if software becomes cheaper, comparatively, compared to generating data sets, software is cheap, right? The software is cheap. You can generate software relatively easily, but data is very expensive. And so what if like only certain entities will be able to build new AI models? What does it mean for research, right? Like what if only DeepMinds and OpenAIs will have the compute power and money to generate data sets? I guess, I don't know. There is like, even if the tools cannot do what they are promising, I think we will probably start feeling the economics of the whole AI paradigm, I guess, fairly soon, if we are not already. And so this is what we are going to need, and I guess this is something that we've heard today, like everyone is trying to generate more data. And again, we don't have a strategy what to do with that data. We still don't know where exactly to put it. We know where to put, structures, PDBs and answer, but there's like a whole lot of other properties and measurements that we just don't know what to do with. And we also don't know what's the best way to get the maximum out of these data sets, very expensive ones, right? And how to optimize them for machine learning. And finally, OpenFold, for example, is a really good example of compute problems. They were lucky. They got like really large donations. They had access to compute, but they often had to like move from one cluster to another, which then required that they kind of adjust their code base to a new cluster. And there are always obviously software, hardware things that you have to work on. But if they had a dedicated resource, I'm sure it would speed them up a little bit. They also have to worry about how many training runs can they run, because each training run is very expensive. We are talking about millions of dollars here. We are not talking about 10,000 or something. This is like very expensive. And so again, which lab, which universities will be able to run, build large foundational models, not just like tweak or fine-tune something that's already out there. And again, what will be the terms and conditions these other models, you will be able to interact with? How will you be able to use them and reuse them? And so I think there is still like hope, so to say, that we will also still need experts. But how do we train experts? And I'm going to go back to Derek's talk again. If we leave a lot of grunt work to machines, how do we learn? Like, we learn through repetition. We also, for us to be also to able to learn and generate knowledge and to like put certain things, like into back of our mind and just be on the autopilot and just be like creative. You have to put like a lot of hours to become an expert. I don't know what's the 10,000 hours to become an expert you have to invest, to master a skill. Not sure what's the heuristics, but if the newer generations kind of lose touch, and if everyone goes to a higher level, like how do we maintain the knowledge? How do we know that how do we get how do we develop our gut feelings if we also outsource a lot of our work to machines? I guess that's I don't have answers to any of it, but these are the things that I'm thinking a lot about. And also, like, what does it mean for us and the institutions and how we work today and how it will affect us? Again, we don't necessarily have to be wiped out because there will be a new, better model that, you know, makes everything we do obsolete. It's just, it's about trends. It's about how people feel. It's about what executives think. Do they think these are the things worth supporting, or will they withdraw their support, right? So it's also about feelings, right? It's not about just what is or how, you know, what the reality is. It's also like what people are excited about and what they want to support, and, you know, and maybe we will cease to exist. And I think that's okay. And the way how I'm thinking about it also, like at the core of our foundation is really trying to provide access, right? Like trying to make things more accessible, more usable, more researcher friendly, and really facilitate collaboration, because I said it's hard. It's not just, you can say, let's just collaborate. Even like when you're at universities and you want to collaborate with another university, then you have to talk to your university people, and then they have to talk to the other university people. It's not easy, it's not simple. There is like we have to come up with new frameworks that really, where, you know, you can say quickly, okay, let's collaborate, and we can be, bang, 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 bang. We can set something up. But for that, it's not just technology, it's also like it's institutions, it's legal frameworks. We have to really think about all of that. And that's why I think technology that we develop, and even the organization itself, is transient, right? Like it's all about what problems are we trying to solve? What is the appropriate entity to actually solve the problem? And, you know, and also what is the appropriate technology? And maybe we'll just have to say, okay, what we are doing at the moment, you know, maybe, you know, we should let someone else or something else take our place and solve the problem. I think the point here with drug discovery, I think we really want to solve those problems. We really want to fight drugs. And it doesn't matter if it's AI or physics-based model or, you know, who does it. Hopefully, someone does it, and we are all better off. And I would like to finish, I guess, again with my hero pigeon, the pigeon. And this is his expression after the disappointing ride, where he says, "You have got to be kidding me," right? And I think even when if we try many things and we fail, I think we have no choice. But when we asked again, we say, "You bet." So that's the end of my talk. I just also wanted to let you know that if you want to connect with us, you can do that through LinkedIn and Blue Sky and X, or you can visit our website, subscribe to our newsletter, which is very friendly. We send it only once a month. It's about 3-minute long read. And again, as I said, please come to Boston to talk to us in person if you have time. And thank you.